Hi, my name is José Hidasi. I'm a biologist, ecologist, and I'm here to present you my academic journey. Okay, so first, why biology and ecology? I'm from a family of biologists, so you can see on the left my grandfather with Helmut Sick, and on the right, I'm here. I'm in my grandfather's museum. So with all that influence, I decided to do biology and ecology, and I chose Federal University of Goiás to start my studies. And in Federal University of Goiás, I studied at Community Ecology and Functioning Lab with my advisor, Professor Marcos Vinicius Sanciaruso, who is a professor in Federal University of Goiás since 2009. And what to do in the lab? Well, basically, we I studied in the lab community ecology and phylogenetic and functional diversity. Community ecology, you know, we study the richness, abundance, and composition of communities. Phylogenetic diversity, we study the dissimilarity of species in relation to their evolutionary histories. And functional diversity is the dissimilarity of species in relation to their biological characteristics. So I'll present you my first paper, which is bird functional diversity and wildfires in the Amazon, the role of forest structure. It was my second scientific initiation, which I did with under the supervision of Marcos Sanciaruso and Jos Barlow. So we already know that wildfires occur in nature. So wildfires are naturally occurring fires in the environment, and wildfires are not very common in the Amazon, but they are getting increasingly frequent, especially when there's an El Nino period. And now, based on niche theory, we can say that composition of species is influenced by species biological characteristics. So disturbances like fire can function as an environmental filter that selects ecologically similar species, or limiting similarity that select ecologically different species. But what does fire do? So fire, in our first hypothesis, would simplify the habitat characteristics, and would make functional diversity decrease. So our hypothesis was that fire selects ecologically similar species, so it lowers the functional diversity of communities. And also, we thought that modified vegetation explains changes in functional diversity, and forest structures related to fire would highly explain changes in functional diversity. We use a model selection for that. So to study that, we got data on forests with different fire histories. We got 10 non-burned forests, 12 burned once forests, and 6 burned twice forests. And these forests were sampled 3 years after the 97-98 El Nino period. And we got some data on the body mass with biological characteristics of birds of the Amazon. Then to calculate functional diversity, we use a matrix of traits, and we transformed it into a distance matrix, Gauer distance matrix, and then functional dendrogram, UPGMA, and then we calculated the NRI and NTI functional diversity matrix. Then We use the model selection process to see how these abiotic variables that you see there are related to the changes in functional diversity. Then we first saw that fire did not work as a filter. Fire did not work as a limiting similarity process as well. But we observed that vegetation structure and functional diversity were related, and canopy cover and understory vegetation density were related to environmental filter, while dead standing basal area and non-woody stems were related to limiting similarity. So what we saw after the model selection process method was that uh, fire would increase functional diversity of species after it modifies the habitat, creating more microhabitats for species to occur on. This was my first paper, Bird Functional Diversity and Wildfires in the Amazon, the role of forest structure, and that was the second time I won the prize for best scientific initiation. That was so good to me. And then in my master's degree, I studied the conservation of functional and phylogenetic diversity. Uh, so identifying patterns and defining priorities. My advisor was Marco Sensaruso, and co-advisor was Rafael Dias Loyola. So we already know that the relationship between human, that there is a relationship between human population and extinctions through time. Uh, as human population increased, extinctions also increased. And species extinction is not random. So it can be selective against body mass or, for example, vulnerability to climate change, uh, species that are easy to hunt, or species that have beautiful feathers. So, and you can see that all three species from the right are from Psittaciformes. And so species extinction can present a phylogenetic proximity pattern. So vulnerability can be related to biological characteristics or functional diversity, and can be related to evolutionary history or phylogenetic diversity. And why is functional diversity important? Because it's closely related to ecosystem services. And why is phylogenetic diversity important? Because it is closely related to functional diversity, and because it is related to evolutionary potential of communities to to create new species and things like that. Okay, so you see that IUCN Bradley's has categories to indicate the degree of endangerment of species based on demographic variables. And but do threatened species have higher functional diversity or phylogenetic diversity than expected by chance? And why to worry about that? Because we cannot conserve everything. We have limited resources and we have to prioritize some species. So that question led me to study my first chapter on my master's degree. It was conservation actions based on reddish do not capture the functional and phylogenetic diversity of birds in Brazil. So we studied scenarios of simulated extinction. For example, we had 20 species in critically endangered category, and we wanted to see if the extinction of these 20 species led to a higher loss of functional and phylogenetic diversity than the extinction of 20 random species. So we could have a higher loss, a lower loss of functional or phylogenetic diversity, or an equal loss of phylogenetic and, functional diversity. And we have different red lists of birds in Brazil. We have a global, which is the IUCN, the national red list, and the regional red list. So the global red list presents 217 threatened birds. The national presents 160 threatened birds, and the regional, which are the six state red lists that we found at the time, present 61 to 216 threatened bird species. So the questions were, does the extinction of birds from threatened categories lead to higher losses of functional diversity and phylogenetic diversity than expected by chance? And does that change in relation to scale? So to study that, we got the four categories that are related to endangerment, to real endangerment in IUCN red list, that are categories that are near threatened, vulnerable, endangered, and critically endangered categories. We got some biological characteristics related to birds from Brazil, to all birds of Brazil, and we calculated functional diversity using the FD index after the creating a distance matrix with Gauer distance and functional dendrogram using UPGMA method. To calculate phylogenetic diversity, we got some random phylogenies from bird tree from Jets et al. 2012, and we made a consensus phylogeny for all birds of Brazil and calculated the phylogenetic diversity using the PD index. So, as I stated before, we used a simulated scenario of extinction to see if the extinction of birds from a category were led to higher losses of functional or phylogenetic diversity than expected by chance. And what we saw for the global, regional, and local scales was that categories commonly presented the same amount of loss of functional or phylogenetic diversity than expected by chance, or lower phylogenetic and functional diversity than expected by chance. You can see in blue that 50% of categories from the IUCN red list showed that species had that the categories presented low functional diversity and phylogenetic diversity than expected by chance. We saw that, you can see that in blue and in black, that in all scales. And we only saw a higher loss of functional and phylogenetic diversity than expected by chance in local scale, when using the local red list. So extinctions are not random. They can be functional clustered, or they can present functional clustering. So birds vulnerable, for example, to climate change, or big birds can make that pattern of functional clustering in red list categories, and they can also have a similar evolutionary history or phylogenetic clustering too. So red lists do not protect higher functional diversity or phylogenetic diversity than expected by chance, and we need for that prioritization methods. So one idea is to use the evolutionarily distinct and globally endangered species, which is the EDGE project, which categorizes species based on how original they are in relation to their evolutionary history. So they can be highly original or less original. But what about the ecological originality of species? They can also be original in relation to their ecological aspects. So our second chapter was to create a new method based on the EDGE project, to identify ecologically and evolutionarily distinct and globally endangered species. So we have problems in biological conservation. We know that some species are higher threatened than others, and we know that there are endemism areas, but we have limited resources, and species are not all similar to each other. So uh, projects like the EDGE project came to prioritize species and areas based not only on how they are threatened, but also based on demographic variables, but also based on evolutionary on the evolutionary history of species. So how does the EDGE project function? It uses the fair proportion method. So, for example, we have those species on the bottom that have their branch lengths of 0.5 and 0.5, and a ascendant species that has a branch length of 4.5. So we calculate the phylogenetic originality of each of these species as 2.75. But how do we calculate that? We get the for each branch length the branch length and divide by the sum of branch lengths until the root, and divide each branch length by the number of descendant branches. So first we have 0.5 divided by 1, then 4.5 divided by 2, and then we have 2.75. We do that for all species, and we have their phylogenetic originality or evolutionary distinctiveness values. So we knowing that, how can we say that which species to conserve? We have a less original vulnerable species, a less original critically endangered species, and a more original critically endangered species. So the first one to conserve would surely be the more original critically endangered species, and that's what the EDGE project does. So it gets the natural logarithm of 1 plus the value of phylogenetic originality of or evolutionary distinctiveness plus the global endangerment, uh, which you can see the numbers here. For example, less concerned is equal to 0, near threatened equal to 1, and critically endangered equal to 4, and multiply it by the natural logarithm of 2. Okay, but we wanted to extend the EDGE project to include ecological distinctiveness. So we wanted to conserve species that are both high ecologically original and high phylogenetic original. So first thing we had to do was to standardize, to put in the same dimensions the functional dendrogram of species and the phylogeny of species. And after doing that, we modified the EDGE index into the EcoEDGE index. So so half of the importance of the index was to phylogenetic originality and half to ecological originality. And to test that, if that makes sense, we use the mammals of the world. Then calculating the ecological originality and phylogenetic originality, we got that, and we can see that there are species that are highly original in terms of ecological and phylogenetic perspective, and there is for example that orange species that is a vulnerable species that has a great, a high ecological originality value and phylogenetic originality value. So it could be a eco-edge species. And so we had three methods to test: the EDGE method, the ECOG method, or ecologically distinct and globally endangered, and the eco-edge method, the ecologically and evolutionarily distinct and globally endangered species. And we have we seeing the maps for the 25% of species with highest uh, index values of these indices. We can see that the Indo-Malayan region is a priority area to conserve, but based on the EcoEdge method, we can see that there's a lot of there are lots of places to conserve to in Africa, South America, and North America. So EcoEdge is a simple method. It is easy to analyze results and can be used to protect a specific function or services or service, for example, related to bee pollinization or things like that. And due to this master's degree dissertation, I got the prize for the popularization of science from the Brazilian Society for the Progress of Science. That was really good to me also. And so I have my PhD thesis, which was the taxonomic, functional, and phylogenetic homogenization, causes, consequences, and conservation implications. So we know that George Cuvier uh, introduced the extinction, uh, the fact that species uh, come to extinction, and there are processes that cause extinction, such as habitat loss and exploitation. But this habitat loss and exploitation, for example, they show selective extinction. So extinction is not random, as we saw in my master's dissertation. Also, uh, Charles Lyell in 1832 noted that some species are becoming more common than others. So domesticated animals present in America were present in America and Australia. And Elton, then in his The Ecology of Invasions by Animals and Plants, he told us that there was going a breakdown of zoogeographic realms due to global commerce. So uh, in general, uh, Mike McKinney and Julie Lockwood, they got this information and said that there was going on a biotic homogenization that was composed of the invasion of generalist species and the extinction of specialist species. So and then Julian Oden, years later, told us that there was a loss of ecosystem functions and services due to that. And why to worry? Because lower phylogenetic diversity and functional diversity is related to higher ecosystem instability. Okay, so we have our three chapters here. The first one is ecological similarity explains relative abundances of small mammals. So we already know that the distribution of abundances of species, that is that few species are highly abundant and then others are less abundant. And there are theories such as niche hierarchy model that say that species arrive and they divide themselves in, for example, the first species arrive and forage on the ground, then other species arrive and forage on trees, and then other species arrive and forage on stems or on branches. Then the species that have the uh, more that are more divided on in relation to what they get from the the habitat, they have less lower abundances. And so but are species are abundant species similar to most abundant species? As we saw here in Sujihara's model, species abundant species are not similar among each other. So to test that, we got the small mammals of the world, and we got we used mixed models to test if the relationship between relative abundances and the functional difference to the most abundant species in each community. And then we put that versus the productivity in each area. We saw that more abundant species are usually smaller, and we saw that abundant species are similar to the most abundant species in communities. On contrary to Sujihara's prediction. And this relationship is mitigated in high productive zones. So areas with high energy availability, uh, species have similar. So we have lots of specialist species working all together. So there is not a skewed uh, relationship. And then we need to know more about the order of arrival of species and the importance of that. Then the second chapter, which is a forest canopy as a living archipelago. Why does phylogenetic isolation may increase and age decrease diversity? So we already know about biogeography of island biogeography, that large islands that are close to the mainland have the higher species richness. So island biogeography can not only be related to oceanic islands, but also we can use living islands like trees. So we have the biogeography of living island or living island biogeography, and in regarding to that, there's a new type of isolation. So a phylogenetic isolation, and for example, if we have lots of pine trees and an oak species in the middle of that, we have a high phylogenetic isolation. So the our hypothesis was that this would work as a type of distance isolation. So diversity would be decreased by this phylogenetic isolation. To test that, we use mites from Oribatida in Forêt de Rennes in France, and we know that these mites present low dispersal ability and low specialization, and we use this index of phylogenetic isolation. I don't want to talk about it right now, but that was the index we used, and we calculated the alpha diversity on branch of mites, the alpha diversity on tree, on each tree, and the beta diversity. So studying the phylogenetic isolation, we saw that higher phylogenetic isolation led to higher alpha, beta, and gamma diversity values. So what we saw was that due to probably to the low specialization of species, they they are really specialized on their trees, but they can also survive if needed on other trees. A pine tree species could go to an oak species. So this uh, arrangement of species could increase species diversity on phylogenetic isolated areas. Phylogenetic phylogenetically isolated trees. Then we have our final chapter, that is climate change will drive species loss and biotic homogenization in a biodiversity hotspot. We know that climate change is happening. It can be due to human causes or natural causes, but it's happening. And we know that it filters a few species that have similar tolerances and work similarly in the environment. And we also know that mountain biomes have low velocity of climate change, and flat biomes have a high velocity, and Cerrado, the Brazilian Cerrado is a flat biome, so it is a good biome to study climate change. And so our idea was that climate change would lower and homogenize community diversity taxonomically, phylogenetically, and functionally. And our model was the Cerrado mammals. So first we downloaded distributions of and climatic variables of the distributions of mammals from IUCN, and the climatic variables from EcoClimate. These variables: annual mean temperature, annual mean precipitation, and seasonality for both. And we calculated the model for the present and a model for the future. Then calculate the diversities on each raster cell and verify future changes. This was the methodology. That was the methodology. To calculate and to do the ecological niche modeling, we use the algorithms of GAM, GLM, random forest, and Mars, and we use the following climatic models, and we made an ensemble of for the future and for the present, and we used these ensembles to calculate a presence absence matrix or raster, uh, if they doing putting an occurrence if the habitat suitability was higher than 0.5. Then we calculated the alpha diversity and also the spatial turnover for and how did we do that? We got each focal cell and calculated the mean of turnover from the focal cell and each of its neighbor cells, and we saw that richness would decrease in no cerrado, and also that we saw that biotic homogenization would be higher in southern Cerrado. You can see that in blue in the Cerrado map. And why in southern Cerrado? Well, probably because the southern Cerrado is the region with the less amount of uh, remaining native vegetation. So what to do then? So we can protect species according to several costs. We can use the eco-edge uh, uh, method and getting the ecological originality of species, phylogenetic originality of species, and red list categories, and get it with land acquisition costs, for example, and then prioritize species and areas, maximizing the eco-edge index and maximizing richness and area size and minimizing economic costs. And to know more about my work, you can go to rfunctions.blogspot.com, and it is a blog where I post uh, everything about ecological analysis. So univariate analysis, spatial analysis, multivariate analysis, functions to do all sorts of things. So go there and see for yourselves that it is a good website. And for example, you can see that there is a function that I created that is bivariate map to create bivariate maps, and also, for example, the eco-edge function to calculate eco-edge index of for species. And also, you can see my two main apps on Google Play that I created. One of them is Cerrado Runner. You use a Cerrado giant anteater to run from the fire, and you could get coins to buy your friend maned wolf. And we have also the app Fábulas Científicas Digitais, which is an app with several scientific fables or digital fables to for you to watch. Okay, so thank you. Don't forget to see my blog rfunctions.blogspot.com, which has more than 700,000 accesses.